Today we're going to be talking about part 1 of our photosynthesis notes regarding ATP and energy. Our learning targets are to be able to identify the differences between two molecules called ATP and ADP, as well as being able to explain the importance of ATP. To start with, think back to the very beginning of the year when we talked about the 8 characteristics of living things. One of those characteristics was that all living things require a constant and continuous supply of energy. And beyond that, we haven't talked a lot about energy and how cells are able to utilize and harness energy to perform cellular work. So I wanted to start with talking about all the different types of energy that we've kind of come across. And this is not an exhaustive list at all. But the first one on my list is heat. And we talked about heat very briefly in our ecology unit as the type of energy that can be transferred from one trophic level to the next, and how it's kind of pretty inefficient because approximately only 10% of the energy from one trophic level is available to the next. And the energy that hasn't been used for cellular work is given off to the environment in the form of heat. Electricity is probably another type of energy that you interact with on a day-to-day basis. Light energy comes to our planet from the sun. And finally, biochemical energy is the type of energy that we have discussed over the course of the semester when we discussed bonds and how different atoms can connect to one another to create molecules. Biochemical energy is probably the one that we are going to spend most of our time discussing today. And really, what does that mean? And how does biochemical energy really work? To help us understand that, I want to show you this graph, and it should look kind of similar to some of the graphs we looked at in our enzyme unit, in which we explored how the energy of a reaction, specifically the activation energy of a reaction, can differ depending on the action of an enzyme. But this graph doesn't necessarily reflect the action of an enzyme. Instead, it's just showing you over the course of time here on the x-axis, so as the reaction is progressing, how much energy is available. And so at the beginning of this particular reaction, we had the reactants, or the ingredients, the things that we started our reaction with. And at the end of the reaction, we have the products, what we ended with. And so based off of the amount of energy, we started with a lot and we ended with a little, which means that there was a significant amount of energy released. So that energy, it has to go somewhere. It doesn't just appear, it doesn't just disappear. So what happens to it? According to our notes, it says when existing chemical bonds are broken, those atoms may form new products that require less energy than before, which is what we're showing here in the graph. The difference in energy results in excess energy that becomes available to the cell, which we indicated here. But what we need to zero in on then is how is that energy transferred, because it doesn't just disappear, it doesn't go away, and it definitely doesn't just appear when it's needed. So instead we have to keep in mind that energy is constantly transferred from the bonds of one molecule to the bonds of another. And this is where it kind of gets into our goal for today when we talk about these new molecules called ATP and ADP. They are going to be the molecules that help us store and transfer that energy until we need it for some other cellular work down the line. So ATP stands for adenosine triphosphate. It sounds like a mouthful, which is why a lot of times we just say ATP for short. But ATP essentially stores and releases the excess energy until it's needed by the cell, and it's a molecule that looks something like this. This is a very basic diagram to kind of show the overall shape of the molecule, but I've emphasized in the name where the T comes from, and it stands for tri. Tri means 3, And when we look at this diagram, there are 3 of these spheres indicated in red, and those represent 3 phosphate groups, as the name implies, triphosphate. If we zero in on that diagram a little bit more closely, we can see that these bonds right here in between the second and third phosphate group are where we're going to store excess energy from other reactions. So when we look at that graph from a couple slides ago and we had excess energy that was released by the reaction, and we said it doesn't just disappear, it doesn't just poof, appear somewhere else. Instead, we're going to use that energy to form the bond between the second and third phosphate group. And so in a way, it's going to just kind of store it until it's needed for later. The other parts of the molecule are indicated here in black. This represents a ribose molecule. It is a 5-carbon sugar, and it is bound to a double ring structure that is very nitrogen rich called adenine. It's a nitrogenous base. Together, when you bond a ribose and an adenine together, you get adenosine. So adenosine is going to be both the adenine and the ribose, and then we have these 3 phosphate groups. So that's your adenosine triphosphate. Interestingly though, if we were to utilize the energy that is stored in this bond and transfer it to a different molecule as a part of a different reaction, we would no longer have 3 phosphate groups, so it would no longer be a triphosphate or an ATP. Instead, we would get a molecule called ADP. ADP stands for adenosine diphosphate. So this is going to be a molecule that's kind of ready and willing to accept a third phosphate group and therefore kind of store away that energy for later. And so ATP and ADP can constantly kind of recycle back to one another depending on the needs of the cell. In this case, as you can see, there's only 2 phosphate groups, where in ATP there were 3. And so the D in this case stands for diphosphate, di meaning 2 phosphate groups. But the rest of the molecule is essentially the same. You still have the ribose bound to the adenine, creating the adenosine. This GIF file just kind of loops through a little bit and shows us how that bond between the second and third phosphate group can be hydrolyzed or broken to release that third phosphate group, which would likely be transferred to another molecule to jumpstart some other reaction. But what's misleading is that this diagram kind of shows the energy released in, like a comic POW symbol. But I don't want you to walk away with the idea that energy is just kind of released out into the open. It doesn't disappear, it doesn't appear, it's always transferred. And so that POW symbol should actually represent a second reaction that is coupled with this hydrolysis reaction. So if we hydrolyze ATP to release that energy, the energy isn't released on its own to go off somewhere. Instead, it's going to be trapped in a different bond. It's going to be transferred. And that different bond that can be formed with that released energy could be anything. And that's the cellular work that can be done by our cell. So this picture shows how those can cycle back and forth to one another. This water molecule is shown here to represent the hydrolysis, or the breaking and splitting of that third phosphate group in ATP. Again, there's your kind of POW symbol to represent that this is a high energy molecule. Once it's hydrolyzed, that released energy can be transferred to another reaction. And then you're left with ADP because there's only 2 molecules left, and that third phosphate has been transferred somewhere else. But we can add that third phosphate group back onto ADP to regenerate a molecule of ATP. And so if there's any other neighboring reaction that we couple to this one that releases energy, we can store it in the bonds between that second and third phosphate group. And so in a way, these can just cycle back and forth as needed by the cell. ATP is a critical molecule to help the cell be able to quickly access and use and store and more or less spend the energy that's needed to conduct cellular work. So this reaction kind of shows how that works. ATP can be broken down into ADP plus that third lone phosphate group, and it releases the energy that was stored between that bond. And again, it's not just released out on its own, it's going to be released to some other coupled reaction. Also, notice the double arrow indicating that this reaction could proceed in the opposite direction to reform ATP as needed to store excess energy. That energy, for example, could be put in to form the bond when building and manufacturing sugars through dehydration synthesis. A common analogy you'll see to represent ATP is a rechargeable battery. So ATP in a way represents a fully charged battery, and as you use that energy and transfer it to another reaction, you're left with ADP. But in the presence of extra energy that has been released from another reaction, it can be used to re-add a phosphate group and recharge your ADP battery into a molecule of ATP. So to recap, biochemical energy is essentially the energy that is transferred and kind of held in place temporarily by bonds within our molecules. Keep in mind that it's great at transferring energy, but it's not going to store it for a really long time. Instead, we talked about our carbohydrates in our biochemistry unit as being ideal for long-term energy storage, which is why in the previous slide we showed a ring structure, which would be just a basic monosaccharide, and that energy can be stored in those bonds until the body hydrolyzes it and breaks it down to release the energy from the food that we eat.